This is Forum, Nature Biotechnology's podcast, where we talk to leading researchers in the life sciences about the latest advances in their areas of interest. I'm Michael Francisco, a senior editor at the journal. Today, on episode 19, the topic is single-cell metabolomics, which gives us a snapshot of the small molecules, intermediates, and products of cellular metabolism within a biological system. Our host is Barbara Chaffee, chief editor of Nature Biotechnology, and her guests are Theodore Alexandrov of the European Molecular Biology Laboratory and Anne Lay of Gigantest, a metabolic analysis company based in Baltimore, Maryland. Barbara, this topic is really interesting. Nature picks single-cell metabolomics as one of 7 technologies to watch in 2023, in all of science, not just the life sciences. Why are people talking about it?
Yeah, great question, Michael. So previously, a cell's metabolome could only be looked at in a whole population of cells or tissues. But the metabolome of a single cell is highly dynamic and changing constantly, as my guest today will tell you. And these lipids, carbohydrates, and small molecules are involved in nearly every cellular process, and they are different in every cell. So recently there have been some technological advances in imaging and mass spectrometry that have enabled scientists to look at the single cell level to begin to understand the extent of this heterogeneity.
Can you tell us a little bit about your guests, Theo and Anne?
Theo is a researcher at EMBL and one of the pioneers who brought this technology to the single-cell level. In 2021, his lab developed SpaceM, which is this open-source software tool that uses light microscopy imaging data to enable spatial metabolic profiling of cultured cells using just a standard commercial mass spectrometer. And Anne Lay is the founder and CEO of Gigantest, which is a company that she started during the pandemic after several years working on cancer metabolism at Johns Hopkins. And Gigantest is one emerging company that is looking to use full metabolic profiles to understand disease and human health.
Great. This should be interesting. Let's get into it. Here's episode 19 of Nature Biotechnology Forum.
Hi, and welcome back to the Forum podcast. Today we're going to be talking about single-cell metabolomics, and I have with me Drs. Theo Alexandrov from EMBL and Anne Lay, who came from Johns Hopkins School of Medicine and is the founder and CEO of Gigantest. We're going to be talking today about metabolites, which are small molecule products that are produced by cells during metabolic reactions, and our metabolomes, which are the collection of all metabolites found in our bodies. Even further, we're going to be discussing the single-cell metabolome, or the metabolites produced by a single cell. So this is a really interesting topic because the cell's metabolome is dynamic and shifts rapidly upon cellular activity and environmental alterations, and it can tell us a lot about the cell's phenotype. So to start off, could you both tell me a little bit about how you got into studying the metabolome?
It happens naturally, actually. Over a decade ago, when I had many projects that required me to have metabolome data to understand cancer metabolism, I had no choice but to master the metabolomics technologies. Once I started seeing what that data could reveal, it stopped feeling like just a tool and started feeling like a whole new way to ask biological questions. That was really the moment I knew this was going to shape the direction of my work. And I found this very powerful and thought I could take advantage of it to answer so many biological questions, not only in cancer metabolism, but also metabolic aspects of many, many other diseases.
Theo, what about you?
I got here by a combination of luck and fortune and everything because I did my PhD 15 years ago in statistics. So something completely unrelated to biology, but I was interested in biology. And then someone invited me to work in this field of mass spectrometry. And back then I've heard something about metabolites, that they are very challenging, the most challenging, what you can get in mass spec. And then I first started working in mass spec. Then I was interested, why all this stuff? And then this is how I got into metabolomics. And I realized that I actually was attracted by a combination of challenge and also promise of this field. And I think this is what we are witnessing right now, that this field really attracts very adventurous people and also ambitious in some way, because there are so many challenges compared to other fields. But on the other hand, and in particular lately, I think there is also a lot of interest and promise in this field, in particular in biology, but also in biomedicine and pharmacology. And I think it's super interesting moment right now to talk about this. So thank you very much, Barbara, for having me. I'm looking forward very much to this conversation.
Yeah, and I definitely want to touch on some of those challenges as well as the promises of this field. But to start off, why do we want to look at this in single cells?
Single-cell metabolomics enables us to do measurements and analysis of metabolites within each cell for many reasons. There is heterogeneity between cells within a given tissue or biological system. One cell's metabolism can be very different even from its immediate neighbor within that same tissue or biological system. And that matters because the cells driving biology are not always the average ones. Sometimes the most important signal is coming from a rare cell population that would disappear in a bulk measurement. So, studying single-cell metabolomics allows us to capture the specific metabolism of that given cell. Because when you study the tissue as a whole, it maps the unique aspects of that cell of interest. Each cell exists in its own dynamic state, with metabolite concentrations changing moment by moment. Even cells of the same type can show dramatically different metabolic profiles depending on their location and microenvironment.
And maybe from my perspective, I completely agree with what Anne said. And let's also think about two examples. One example is fertilization, and another example is immune system. So this is where we can see that one cell can really make it or break it. So, and digging a bit deeper into immune system, which is like, I believe is one of these promises. If we start thinking about, cells there, which are in the organism or maybe introduced in cell therapies, and we can start imagining how one cell can proliferate and give actually rise to clonal population of a lot of functioning or maybe dysfunctioning cell, right? So this is where it all started. It starts to really matter on a very practical terms. But another very, very interesting aspect with single cell analysis that we have learned from single cell biology and single cell RNA-seq over the past 5 years, not only that every cell is unique, but also if we start considering analysis on the single cell level and not just bulk, we can actually uncover a lot of systematic relationships and almost like new mechanisms, besides just new cell types, of course. So those mechanisms that we cannot really see behind this world of averages. And this is the same expectation and the same promise we currently have in single cell metabolomics.
So you've both touched on this idea that, single cells are so heterogeneous, and when you're looking at full tissues, these results are masked. So yes, the single cell is going to tell us so much more detail. And the metabolome is really dynamic, right? It's far more dynamic than the genome or the transcriptome. So this has to have some challenges for how you go about studying these communications, these processes. What are the main challenges here in looking at the metabolome, especially when we're looking at single cells?
But if you allow me, before we go into the challenges, I want to add a very important aspect of why we study single-cell metabolomics: disease diagnostics and treatment, especially in the drug discovery field where you can tailor specific therapies. To me, that clinical angle is what makes this more than a technical achievement. If we can read metabolism at that level, we can start matching treatment to what the disease is actually doing, instead of treating every patient the same way. Of course, you want to target the disease metabolism specifically. The good news is that disease metabolism is very, very different from normal cell metabolism. So if you can understand the difference between disease cell metabolism and normal cell metabolism, you can design very specific therapies that help tremendously. Think of it as the concept of a smart bomb. You don't want to bomb the whole tissue, including the innocent normal cells or cells that are not in the disease state. This precision is what makes single-cell metabolomics so transformative for drug discovery. By targeting only the dysregulated cells, we can dramatically reduce side effects and improve therapeutic outcomes for our patients.
And I think Anne is making here an excellent point, that it's not only about heterogeneity, but we know that some phenomena in disease in particular, they actually can be manifested or detected only through a rare population of cells. And one example here would be circulating cancer cells. And we know that from the amount of blood that can be actually taken from a patient. So sometimes it's possible to detect or isolate, or even have the presence of only a handful, maybe a few tenths of circulating cancer cells. And in order to actually to detect them, but then also understand their metabolism or metabolome, of course we need here single cell metabolomics, right? So I think we should not forget that already proven presence of these rare events that sometimes, in particular in disease, or maybe early diagnostics of the disease, can be extremely important and can be also make it or break it for predicting the health and maybe a therapy or therapy selection of a patient.
Yes, absolutely. I absolutely agree. And finding these rare cells, obviously, is a challenge in itself. So, yeah, let's get back to those challenges. And so what challenges do you see are, I guess, the most significant for looking at this at the moment?
So the great thing about single-cell metabolomics, as we already mentioned, also creates a lot of problems. You know, in order to have, as the term says, a cell, a single cell, you have to go through a process of isolation to obtain one single cell. Either you study it directly, putting it into the mass spec, but that process itself has a side effect. You change the environment, the tissue environment, the biological system environment. That single cell may behave very differently in the isolated state versus when it has its neighboring cells because of cell-cell communication. So the data that comes from single-cell metabolomics may not reflect to the actual metabolism of that cell when it was in its natural setting with neighboring cells in the biological system. Because, you know, your metabolism this morning before breakfast is different from after breakfast, let alone a cell with thousands and millions of other cells around it with the true phenomenon of cell communication, and then the metabolism measured after the artificial process of isolation. That's a human limitation, because nobody can instantly go from tissue to single cell. No matter how you do it, it takes time and creates an artificial environment. So, even with better instruments, we still have to be humble about what the measurement represents. The question is not only whether we can measure a single cell, but whether we can preserve enough of its original context to interpret that measurement correctly. In biological systems like serum, plasma, urine, CSF, organs, or tissues, there is no such thing as a single cell per se. They all live together. That is the biggest challenge to me. This is why method development is so critical in our field. We are constantly refining isolation techniques to minimize the time between extraction and measurement. And we are developing new approaches that can capture metabolic states with minimal perturbation. Even small improvements in sample preparation can significantly improve the quality and biological relevance of our data, which ultimately leads to better clinical insights and better outcomes for patients.
And I completely agree with you. So I think this dynamic nature of metabolism and also us wanting to avoid unwanted perturbations, of course, is one of the key challenges. But also to put it in a bit of high perspective, I think first and foremost, this is actually amount of material. Because just to put it in perspective, we also run in so-called bulk metabolomics services here. And for bulk metabolomics services, if I go to our scientific officer, they ask us 1 million cells, right? So, and this is what the field was about, right? To do experiment, you need 1 million cells. And what we're trying to do right now, and what we actually have succeeded as the field, to get reasonable, informative readouts from 1 millionth of this amount, right? So this is, of course, this is challenging. It requires pretty much change and build up from scratch everything. Another challenge, which might be not that obvious to some biologists, is the lack of amplification. In sequencing, we have learned to rely on the possibility of amplification of major molecules like DNA and RNA, right? So in order to increase the detection, this is why PCR works. This is why we can do SARS-CoV-2 detection in just like with a simple like PCR test, right? Very sensitively. So here we actually unfortunately lack this capacity to amplify the metabolites before we detect them. And then we also should actually start thinking about what is this metabolome? It is in the cell, but start thinking about this, a lot of small molecules, they freely diffuse through the cell membrane. That means we want all of them to be there, because we lose part of them, we actually lose the detection. But at the same time, I mean, if they start like really diffusing out, we need to be with our cells as gently as possible. We don't want to stress them, but we don't want the metabolites to leak. So, and these all aspects which make it also that in single cell metabolomics, we need to rethink completely how cell prep is done and start from ground zero. And the last thing is also, it comes from the key phenomenon of metabolism, that it actually captures non-genetic factors. And I think Anne has alluded into this. Diet is a huge portion, but it's not only diet, right? It's our exercise. It's actually like, do I have now sunlight or not, right? This all affects, and here we, at some point, we can detect a lot of molecules, non-genetic factors, diet and everything. But we actually do not know how they're produced, how they're interconnected, and how they're also metabolized by our cells. So accounting for all these non-genetic factors brings us to one of the problems and the challenges, which is called metabolic dark matter, where there is like, the metabolomics produces so much data, but it's so hard to actually annotate them, pinpoint them to individual molecules, and make sense out of these molecules. So all this creates like this fascinating but also challenging field, and the challenges that we're actually facing in the single cell metabolomics field.
I want to wrap up so that the audience can understand the challenges. Theo and I, it's interesting that he mentioned the challenges of obtaining the data. Single cell metabolomics is very challenging because, usually people work with, as he mentioned, 1 million times more cells, and now it's down to 1. Getting the data is a technical challenge. I'm talking about another aspect. Even if you have the ideal technology that gets you the metabolome of a given single cell, keep in mind that the data of that single cell is not the actual metabolism of that cell as it was in the biological system, because of the artificial process, as I mentioned earlier, of isolating them into single cells before you can begin the assessment of the metabolome of that cell. Make sense? These technical limitations should not discourage us, but rather motivate further innovation. Every new technology advancement brings us closer to capturing the true cellular metabolic landscape. The field is evolving rapidly, and what seemed impossible just a few years ago is becoming routine today.
Yeah, exactly. So, you have the issue of scaling to just one single cell, but also you're taking that cell out of its native environment. So, and Theo, you talked a bit about this dark metabolome. So these are the fraction of peaks you get from these experiments that can't be assigned to any certain metabolite, right? So what is needed there to shed some light on what these peaks are?
Yeah. And just to put it in perspective so that people will understand the gravity of this problem, we are using technology called imaging mass spectrometry. So we put a cell, so tissue section, into this instrument, and it generates 100 gigabytes data. Out of this 100 gigabytes data, right now, we can find information and localization of, let's say, between 100 to 500 molecules. 100 molecules, images for them, definitely we do not need this 100 gigabytes. It can be easily packed in probably 1 megabyte, maybe like 10 megabytes. So we're just scratching the surface of this wealth of data which we're generating. So this is what normally people refer to dark matter problem in metabolomics. So what's needed? If I knew, we would probably be solving it already. We actually do. We are solving. But I think we need to have, first of all, we need to better understand the fundamentals of molecular ionization, or how the signals in mass spec come from like material from the tissue. Absolute fundamentals of analytical chemistry. This is one. The second, what we need is to have better computational methods that would be modeling how this process is actually going and how is it affected. And also what we see is that we need completely fundamentally new computational approaches, because the approaches which exist right now, they pretty much give only incremental improvements. So that's why there is a huge hope for machine learning. And what is now, in particular in the era of ChatGPT, everyone is talking about generative models, right? The models that will be generating words. Can we now create models that will be generating how the signals, how they come from molecules or from cells? I mean, at some point, if we get to this point, then this will help us also understand which molecules were in the first place over there. So there is a huge promise and hope for machine learning to help us to chip off more of this dark matter and get more information about more molecules which are actually represented there.
Yeah, that's a great point. A lot of what you're saying here kind of reminds me of challenges in the single-cell proteomics field as well. You know, with the scaling up and with the amplification and, you know, the potential use of these machine learning models. So do you see any specific differences that could be noted about when you're looking at metabolomics versus proteomics, whether in the methods or the challenges?
Before we go into the comparison between metabolomics and proteomics, I want to add some more aspects of metabolomics as we see it today. We have become much better at listing what is high and what is low. The next step is explaining where those changes are coming from and what is driving them biochemically in that specific person. You see high-low comparisons of one metabolite or even thousands of metabolites, but that's the reason Gigantes was born last year, 2022, because we want to bring metabolomics to the next level. I congratulate you, Theo, for your skill in computerizing the high throughput data. From our angle, we want to chip in on the biochemistry aspect and tackle the next level of metabolomics. Why we had those glucose and cholesterol levels. Because, you know, human metabolism is extremely complex, but it's actually very simple. Any biological system, whether animal or human, metabolizes vitamins, minerals, carbohydrates, protein, and fat. So when someone has high cholesterol, the real question is, where is it coming from? Is it from carbs? From fat? From protein? Most tests today can only tell you, sorry, your glucose is high, or sorry, your cholesterol is high. They can't tell you why. Gigantes goes a step further. Using our proprietary technology, we can trace where each metabolite comes from and how it is produced in that specific person, which is what makes therapy actionable and targeted rather than generic. That's the level Gigantes is bringing to the field, turning data into a real, personalized plan. This is why understanding the source of each metabolite is so critical to making informed clinical decisions. Without knowing where a metabolite originates, we cannot design effective interventions. The future of precision medicine depends on this kind of detailed metabolic insight. By tracing metabolic pathways back to their nutritional and biochemical sources, we can create truly personalized treatment plans. This shifts metabolomics from being just a diagnostic tool to becoming a powerful framework for actionable health interventions.
That's a great point. I mean, and we're going to shift shortly into the applications here for this tech. I know there's a lot to say there. To just, I guess, get back to the question about comparisons to proteomics, do you have anything, or is it the same? Or are a lot of the challenges just that? Are they the same?
About proteomics and metabolomics, I want to emphasize one thing. You may have high expression of a given enzyme protein. The gene makes the protein, and enzymatic proteins catalyze metabolic reactions, which dictates whether a metabolic reaction happens or not. But often we observe that even if you have high expression of the given enzyme protein, the metabolic reaction does not necessarily happen. So when you see, for example, high levels of lactate dehydrogenase A or B, it does not necessarily mean that you have high levels of pyruvate being converted to lactate. But when you see high levels of lactate, you know with certainty that the reaction did happen. It's not that because I'm in the field of metabolism, I'm saying one field is more important than the other. That's not my message. But I want to say that studying metabolism relies heavily on measuring actual metabolites rather than the protein that catalyzes the metabolic reaction. Because sometimes it's not 100% guaranteed that even if you have high expression, the metabolic reaction will happen.
Yeah, the questions being answered are different, right, when you're looking at proteins versus metabolites.
Yes, and I think Anne alluded very, very well into the problem of metabolic plasticity, which makes so hard to interpret the data. But I think we also have comparing to proteomics on technological level, right? Even before we get to the point when we actually have the data to interpret. So there are several interesting aspects which distinguish metabolomics from proteomics. And one is the structural diversity of metabolites. And here you might say, oh yeah, there is huge structural diversity of proteins. Let's look at AlphaFold and all the potential structures which are currently being generated somewhere on Google servers, right? So, but the thing is that, of course, huge diversity of proteins as well. But for metabolites, it's not only structural diversity per se, but how structural diversity will be changing the ways how they can be detected. And then this actually gave rise to a situation where in metabolomics and single-cell metabolomics as well, there are multitudes of protocols and methods for detecting this group of metabolites, that class, this pathway, and you can never get all metabolites in one go with just one protocol. The situation that we are now currently have in proteomics, where it's pretty much the same protocol. And again, there is incremental development improvements, but mainly action computational data analysis right nowadays. So in metabolomics, it's almost like Wild West. You go there and almost every day or every week there will be a new protocol developed because everyone tries to improve this detection. And this also, I think that there are multiple challenges there as well because the structural diversity is almost unlimited. And I think Anne alluded into this nicely by saying that metabolites, they get modified not only through the enzymatic reactions. Of course, this is a major part of metabolism, but we know that these reactions can be spontaneously happening. We also need to account for those non-genetic factors and think about vitamins, right, in our body. So how they get in or how they don't get in, and then how it all propagates. And it also actually creates sometimes even computational challenges that I know well because I work in this field. When you want to annotate or to do metabolite identification or protein identification, there is this approach where you need to give into this approach so-called unfeasible or impossible entities or proteins, for example. You do it by shuffling the sequence. In metabolomics, almost any structure is possible. So even how to think of impossible metabolites might sound like, a very abstract problem, but for computational developments, it's not. So long story short, this almost infinite structure diversity is a challenge. And once I've heard the saying that if you take a drop of seawater, you will find almost any structure there, structure of small molecule. Because think about this UV radiation, which breaks also the molecular chemical bonds, creating new structures. So this is the problem that we are facing. It's absolutely not abstract, and this is what makes metabolomics so challenging.
Those are great points. And what's amazing to me is that there are all of these challenges, and this is such an emerging field, but we still have companies that are being formed to use metabolomics to treat diseases, to look at cancer, to look at just general human health. And to me, one of the first applications here that comes to mind is, looking at cancer and looking at tumor microenvironments and immune cell metabolites. We know cancer cell metabolisms change, and this could influence immune cells. We can potentially influence immune cells, right, to have more kind of functional metabolisms. So could you guys maybe touch on what's happening in the cancer space currently using metabolomics, single cell metabolomics, and what do you find exciting there?
If you talk about a specific cancer as a disease, it involves the gene mutation that the person unfortunately has. There is a famous statement that cancer is a genetic disease, but recently we showed that cancer is also a metabolic disease, because it was proven by a Taiwanese group who published a very famous paper in which they exposed innocent pancreatic cells to a high glucose environment to mimic diabetic patients. And guess what? The innocent pancreatic cells developed the novel KRAS mutation and became pancreatic cancer cells, just because they were chronically exposed to a high glucose environment, similar to what diabetic people experience. That is why I do not see metabolism as a side story to genetics. It is part of the cause. And in some settings, it may be the piece that explains why one person progresses differently from another. So this is an application that we have to see the big picture. Not only the mutation leads to the disease, but also the metabolism. If you talk about Parkinson's, you talk about schizophrenia, which has recently been linked heavily to genes. But once you have the gene, the actual outcome, metabolism, is the final determinant of how the body reacts to internal factors. In this case, genes, proteins, or external factors like the way we eat, the diet, the lifestyle, the environment, pollution. Take red meat and smoking. You hear all the time that it's not good and that it increases the probability of getting cancer. But there are people who smoked all their lives and never had cancer. And there are people who never smoked a single cigarette in their life and unfortunately have cancer. So apparently, smoking just pushes the probability higher. It's not 100% guaranteed that you will have any particular disease. The metabolism of that person will react to any given factor, smoke, environment, diet, and make the disease happen or not. Not solely the intrinsic or extrinsic factor. Human metabolism is the final determining factor, because in order to make genes, you need A, T, C, and G. In order to make proteins, you need amino acids. Metabolites are the fundamental elements of life. The gene people may get upset with me because they say their genes are the fundamental elements of life. But how can you have a gene without A, T, C, and G? A, T, C, G are metabolites to help genes replicate rapidly and make new cells. Also, in order for new cells, you need lipids. How can you have a new cell without a cell membrane? The major components of the cell membrane are lipids and so on. My wrap-up sentence about applications is, we have to look at the big picture. from both intrinsic and extrinsic factors, and how the biological system reacts to these factors and actually develops a given disease or not.
Yes, and from my perspective, with respect to cancer metabolism, I mean, first and foremost, I'm here more of external observer because I'm not really coming from this field, but it is absolutely fascinating for me to see how researchers and scientists in this field, how they're getting closer to understand the role and the function of this, what is discussed as hallmark, right? Reprogrammed cancer metabolism, reprogrammed metabolism in cancer cells. So, and how also similar reprogramming is now getting revealed in other cell types as well. And now we start thinking that, okay, how this is really, metabolism is intricately related to the disease, but also to the environment. And Barbara, as you said, like tumor microenvironment, when you start thinking about this, it is absolutely innate. It's a foreign environment for the cells, right? It is highly acidic, highly hypoxic. And we can start thinking about Warburg effect, where the cells rely on glycolysis. So as adaptation to this environment. And for me, what's the most interesting direction which is now going on is to really understand the competition between cancer cells and immune cells in this environment. Because I think understanding this will make us one step closer towards creating more effective therapies, in particular cell therapies and adaptive cell therapies, to really reprogram or support the immune cells, even from our organism, to really fight the cancer where it's happening. And here I just would like to give a huge shout out to such researchers and scientists as Erica Pierce, actually currently Johns Hopkins as well, who pioneered this notion of metabolic competition and also metabolic requirements. Because if we start thinking about the systems, so the cells in the tumor microenvironment, they need to have metabolites not only as energy, but they need them also as building blocks for proliferation and so on. But also some of them, because metabolites are so ubiquitous, they actually potentially evolved to react to abundance or to absence of metabolites, perceiving them or sensing them as signaling molecules. And it's absolutely fascinating to see what's going on right now in the cancer metabolism field beyond just Warburg effect, So glycolysis versus oxidative phosphorylation, right? So I think this is really, really exciting. There is so much more going on. And immunometabolism is definitely this frontier, which will be shifting the needle in our understanding and hopefully also fighting cancer.
That circles around really nicely to what Anne just said about how these metabolites are just the basis of how our cells interact, and understanding that at that level is really significant if you want to go into any kind of treatment or therapy. What other directions do you see this research going in, whether it's towards human health or towards other diseases or conditions?
I want to add a little bit about the word hallmark of cancer. The hallmark here is not only the ABCD metabolites that are signatures of cancer. For example, the Warburg effect for cancer. It's not just the name of a metabolite as unique in one disease and absent in normal conditions. Because now we realize we have an article about the metabolic interplay between cancer and other diseases. The Warburg effect is not only happening in cancer, but in other diseases as well. So it's about the way a disease reacts and develops in response to internal and external conditions. If you grew up in America, you may know the game called whack-a-mole. So with cancer, we call it whack-a-molecule. You suppress one pathway, and it has a backup pathway. It has a way to escape therapy, grow perfectly fine, and relapse. So, the hallmark understanding in the broader term is how that specific disease reacts to therapy, reacts to the environment, and whether the unique metabolism of that given patient is resistant or sensitive to therapy. Not just a very simple list of hallmarks, because nothing becomes uniquely defined until we study it further and further and say, oh, by the way, the hallmark Warburg effect is not just for cancer. We saw that in frail subjects versus young subjects as well.
Good point.
And from my perspective, in terms of probably the most promising applications, and in particular the focus on human health and disease, because of course in biology there will be so much revealed, I think now with these technologies available, but if focusing on human health and disease, so I really hope and to see how single-cell metabolomics can help in 3 areas. The first, I think, will be drug development. And here, even just to understand where does a drug go, how is it metabolized, and actually what does it change, specifically in situ, with respect to metabolism, and also how the adverse metabolic effects are created. So because this is one of the challenge to reduce metabolic toxicity of the drugs and drug candidates. So I think this is what we'll see in drug development. I think the next one will be probably therapy selection or therapy monitoring, because here metabolic profiles, they're so sensitive, and they also at the same time they are preserved. So that means I really hope that we'll see new developments in, first of all, probing different therapies. And for example, some work we started already, and others I know also working on this using organoids, where you can subject organoids to different therapies and then pick that one which has the best metabolic effect. So, but on top of this, I think in terms of therapy development, probably cell therapies has the biggest promise for single-cell metabolomics, because there is now already enough evidence to link the immune cell function with metabolic state. So if we can actually optimize the adoptive cells therapies, not only genetically, but also metabolically, and the work has already started, then we will make them more fit to actually fulfill their function in vivo after they actually inject it. And then I think we'll see also diagnostics, because metabolic biomarkers, they are real, they are used already, but now the question, can we actually adapt and make diagnostics better using liquid biopsies? I think so. Because, cells, they are informative for the state of disease or therapy response. And last one, I think that at some point we'll actually capitalize on undiscussed yet aspect of single-cell metabolomics that is the cheapest and the fastest of all single-cell omics. So this opens so much possibilities for us to generate a lot of metabolic atlases and feed them to some AI that will be actually generating structures which have minimal metabolic effects or minimal metabolic adverse effects. And this will be very, very exciting, because this will make overall drug and therapy development faster, cheaper, and actually less toxic or causing less adverse effects that we are having right now.
Because you mentioned immunometabolism, the most important part is that we try to understand immune cell metabolism as much as we try to understand cancer cell metabolism. So we have 2 opposite directions. We want to compromise the cancer cell metabolism, but promote, help, and nourish the immune cells to fight the cancer cells. That is where this becomes hopeful for patients, and not just interesting for researchers. The goal is not simply to attack harder, but to attack more intelligently while preserving the cells that help the patient recover. The old-fashioned was that you just give cisplatin and it kills every cell, and the person loses their hair, loses weight, and a ton of other side effects, the typical image of cancer patients. Because, unfortunately, the side effects are on every cell. Every cell was killed. So, as scientists, we impose this requirement on ourselves that we have to understand single-cell metabolism in order to hit the right enemy and protect our allies. Kill the cancer cells, but nourish the good ones so that the patient has a good quality of life and uses their own army to fight the cancer cells. It's doable. It's doable with single-cell metabolomics. We can get there. We are making great progress, and we will get there with technology, awareness, and the willingness of scientists in this very promising field. This dual approach represents the future of cancer therapy. By understanding both sides of the metabolic equation, we can design treatments that are both more effective and less toxic to patients.
I think that's a really great note to close on. So thank you, Theo and Anne, for sharing your perspectives on this topic and also for your research and work in this space. I think it has so much promise, and I'm really looking forward to seeing what happens in the future. It's been a really interesting conversation, so thank you both.
Yeah, thank you, Barbara, for inviting us and me. And it has been a pleasure. And thank you also for picking this topic. And I hope that the word will spread further, that this technology and this field is emerging, and we're very much looking forward to it.
Wonderful. Thank you, Theo. Thank you, Barb.
That was episode 19 of Nature Biotechnology Forum. Thanks to our guests, Theo Alexandrov and Anne Lay. You can listen to all episodes of this and our other podcasts by searching Nature Biotechnology wherever you find and listen to podcasts. If you have any thoughts or comments on our podcast, please tweet us at Nature Biotech. That's all for now. Until next time.